Hello and welcome to this new Gen webinar sponsored by Carterra, entitled The Therapeutic Antibody Screening Revolution in the Post-Genomics Era. I'm Jeff Bogaliskas, Technical Editor for Gen, and I'll be the moderator for today's event. Delivering efficacious therapeutic candidates involves screening your entire panel to understand their kinetic and epitope profiles, while understanding each candidate's relationship relative to others. In combination with other data, such as antibody sequences and epitope mapping, high-throughput epitope binning and kinetic screens enable fast and optimized selection of antibody panels for mechanism of action studies, while simultaneously providing a crucial evaluation of antibody generation technologies. Excitingly, this new technology has been applied over the past several years by companies and institutions in order to advance their therapeutic antibody drug discovery programs for cancer, infectious diseases, heart disease, and many other therapeutic areas. Increased throughput coupled with unique epitope and kinetic analysis software has disrupted the status quo of antibody screening and characterization, advancing monoclonal antibodies, bispecifics, antibody drug conjugates, and CAR-T therapy programs. Now let's meet our presenters for this webinar, who will discuss how a modern-day high-throughput surface plasma resonance, or SPR, has facilitated a paradigm shift in antibody screening, enabling higher information content assays to be conducted earlier in the research pipeline in order to streamline lead selection. Dr. Zimpel Matharu is a scientist at Bristol Myers Squibb, who is an expert in biosensors and has co-authored approximately 30 peer-reviewed journal articles in the area of biosensors and surface chemistry. Today, Dr. Matharu will discuss how characterization of large antibody panels is shaping lead identification and how Carterra's LSA platform is aiding in the screening process. Noah Ditto is a senior application scientist at Carterra, where he helps provide hardware and software solutions that enable high-throughput SPR analysis in therapeutic drug discovery. Mr. Ditto will tell us more about high-throughput SPR and the specifics of Carterra's automated screening platform. However, before we move on to the presentations, I want to remind the audience to submit questions for our panelists for the Q&A session that will come up right after the last talk. It's very simple. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. We'll try to get to as many questions as we can. All right, with all the logistics out of the way, let's get our webinar started and turn the presentation over to Dr. Matharu.
Hello, everyone. Thank you for joining today's webinar. Today, I'll be talking about how early characterization of large antibody panels reshape lead identification and technology development process. Well, in the early drug discovery process, a large number of antibodies are generated for a specific antigen, and finding the antibody that targets a specific region on the antigen or epitope is highly crucial, because epitope is an innate property of the antibody, and only particular epitopes influence the function of the target protein. So getting the epitope information upfront for all the generated antibodies along with their kinetics and affinity information can allow us to sort the right leads and choose an optimal antibody panel. However, the limited throughput, long assay time, and high sample consumption of the existing binning methods enabled the study of only small panel of antibodies. As we could not characterize all the generated antibodies, we might have lost some very interesting epitopes in the process. In this context, the LSA allows a full symmetric binning of up to 384 samples accompanied by affinity determination. As you can see on the slide, that it has a multi-channel printhead to create an array of biomolecule, and it has a single channel mode for analyte detection. Well, I'm not going too much into the detail about the instrument. I'm sure Noah in the next talk will talk more about the instrumental capabilities. So in my talk, I'll be focusing on how this instrument helped us characterizing a huge set of antibodies coming from different sources and how it reshaped our selection criteria and helped us to pick the right hits. For any specific antigen at the BMS, we receive antibodies from different sources. BMS has been a pioneer in engineering different transgenic mouse strains, and knowing the performance of each mouse strain is very important for us. So we receive antibodies from different transgenic mice via different versions of immunogens. The most common one is the recombinant soluble protein. We receive antibodies from different routes of immunization and different dosing regimens, and also very important from different antibody generation techniques such as traditional hybridoma, B-cell encapsulation, and single B-cell cloning, and even from different people, where some people select with a wider net and select hundreds of antibodies for an antigen, while some people throw a narrow net. So for this particular antigen, we force tested about 600 hybridoma supernatants on the LSA via capture kinetics, which is a quick way of investigating the binding kinetics of unpurified samples. So as you can see on the slide, the cartoon, we made a lawn of polyclonal antibody on an HT-30M, the low density chip provided by Carterra, and then captured the antibody supernatant, and then studied the antigen binding on it. So this slide shows a small subset of the binding sensograms we obtained via capture kinetics. As we can see, a very wide range of antibody affinities. We have antibodies with very high affinity, and we have antibodies with medium or low affinity as well. Well, although capture kinetics is a nice and quick way to study antibody-antigen interaction, but it is probably not the best for a long binning experiment where one has to capture the antibodies in each binning cycle, regenerate, and then capture again. And also a blocking step is needed when you want to run a binning experiment in the capture format. So in that case, purified samples can be very helpful as they can be covalently coupled to the sensor surface and they can provide a better surface integrity. So later we used purified set of antibodies that contained a subset of the purified samples from the previous hybridoma samples and some additional samples from the other antibody generation techniques such as B-cell encapsulation. So this set was a very large set. So as you can see on the slide in the small cartoon on the left, that we covalently couple these purified antibodies onto a HT-30M, the low density chip again, and perform the couple kinetics on the LSA. And the left-hand side is also showing the subset of the binding sensorgram we obtained from the couple kinetics. And the good thing about coupling the antibodies to the surface is that you can use the same chip for a binning experiment, as can be seen on the right-hand side figure of the slide, showing the raw binning data of the coupled antibodies in a classical sandwich format, where the first curve is showing the antigen binding and the second is showing the sandwiching antibody step. In order to validate the instrument and also the quality of data we are getting on the LSA, we compared the kinetic parameters of the overlapping antibody subset, which we had in the unpurified soup form and then later in the purified form, and we tested it via both capture kinetics and couple kinetics. So the result is shown on the slide. So you can see the comparison on the left. Except for a few outliers, we saw a good correlation between the KD values from the capture and couple kinetic assay. Though we are not comparing apples to apples here, the formats were different in the two assays, but despite of the differences, it was still in a good agreement. To further validate the instrument, we also compared the capture kinetic data on the LSA with the capture kinetics data on the high-throughput Biacore instrument, the Biacore 4000 instrument. And again, we saw a very good correlation in the data. While the off rate was a little bit faster on the Biacore 4000, the extreme right figure here, which can be due to several reasons, one of which can be the differences in the sensor chips we used for the two assays. For the Biacore experiment, we use the CM4 chip, while for the LSA experiment, we use the HT-30M chip provided by Carterra. And not to forget that it took about 2 to 3 hours on the LSA to test 384 antibodies via capture kinetics, but on the Biacore 4000, it took more than 50 hours, about 50 hours, with several fold more sample consumption on the Biacore as compared to the LSA. So next we use the same antibody coupled chip for the high throughput binning experiment. And before I discuss the binning data, I like to mention that running a quick kinetic test before binning is very helpful to find out the good binding samples, the good binders, the non-binders, and their heterogeneous sample. And so you can exclude them if you don't want to include them in your binning experiment and scale down your panel a little bit. Well, the binning results are generally visualized in the form of heat map, as shown on the slide, where the vertical direction depicts the coupled antibody on the sensor surface, and the horizontal direction shows the sandwiching antibody. And this was the high throughput heat map we got on the LSA, showing the binning results of about 288 antibodies coupled to the sensor chip against about 45 reference antibodies or benchmark antibodies. These reference antibodies are the clones we were most interested in, or the control antibodies, or the antibodies from various patents. And this binning goes a long way. Interpreting the relationship between the antibodies, investigating their blocking, non-blocking profiles, their epitope diversity from this giant heat map can be quite scary. So the LSA software provides you simpler ways to visualize the same data. Via some of the nice features of LSA, we converted the same data into the form of network plots or community plots, which helped us sorting the antibodies into groups based on their epitope diversities. And this figure on the slide is showing one of the simplest walk trap visualization of the LSA software, dividing the same data into two big groups. Well, the walk trap visualization is not sufficient to understand the full diversity of the sample set. So it makes sense to go into some more detail, like on this slide. Here we are looking at the community plot where the samples are divided in 6 major groups. With some individual samples, which does not belong to any big community. Well, I should mention that the communities show samples with similar behavior in the epitope binning, but the behavior doesn't have to be completely identical. In order to analyze the data in different ways, the LSA software allows you to color code the samples into in the community plots based on different parameters such as affinities, mouse info, antibody generation techniques, etc. So if you have all that information, you can analyze the data in a better way by color coding the community plot. So first we color coded the plot based on antibody affinities as seen in this figure. And this was very useful because once we knew the epitope diversity, we could pick good affinity antibodies from each group. And here, different colors are representing different range of affinities. For example, anything that is close to the red color is high affinity, and anything that is gray is either very weak or heterogeneous. Well, if we look at this plot, there are some individual samples that we saw in the community plot, and they're very weak when we color coded based on affinity. So we kicked these samples out because these samples can give you false negatives. They can also separate the related samples in different groups. And this is how it looks like after removing those extra samples. We also looked at highly related samples to QC our binning assay. For example, the black squares on this slide are the variants of the same parent antibody clone. They are all binned together in the same community, and they are showing identical blocking, non-blocking profiles. So it is providing a good QC for our binning assay as well, that how the related samples are behaving. Are they showing the identical behavior, or they're not showing the identical behavior? Well, it should be noted that when we removed those weak samples earlier, the community plot remained intact because that depends on where you put the threshold or the pruning line on the dendrogram to generate the communities. And the dendrogram is another visualization that the LSA software provides, and it's highly helpful to spot redundancies within the sample subset that I'm showing on the next slide. In the combined dendrogram view, you see the pruning line in red, where it is said to overread the minor differences when creating the community plot. However, even at the highest level of detail at the bottom, you can see a group of samples showing exactly identical behavior. Later, we found out that these samples had highly related sequences. Well, it can be a good QC for a binning experiment that the replicates are showing identical behavior, but we would like to avoid redundancy within the sample subset in future. So we gave the results as a feedback to the selection technology people and advised them to change the selection criteria. One of the very interesting thing about this program was that the antibodies were generated from several different transgenic mouse. As I mentioned at the very start of my presentation, and we wanted to know which mouse strain gives us the best antibodies. Like here, when we color coded the community plot based on 7 different mouse strains, the different colors of the mouse, the different colors at the left are representing different mouse strains. We observed that the red mouse strain is producing very high diversity in the sample subset, as it is well distributed in all the communities. You can see it on the slide. While if you look at the yellow mouse strain, despite of having a large number of samples, it's only distributed in 4 communities. Next, we compared how different mouse strains correlate with different affinities, and it was very interesting to see that the red mouse strain is not only showing high diversity, but it also producing high affinity antibodies. As you can see on this slide, the left-hand side figure is the color coding with the mouse strains, and the right figure is the same community plot color coded based on different affinities. So if you look at the middle group, the red mouse strain is on the right producing high affinity antibodies as well. So getting all that information upfront in a single experiment really helped us to see which mouse strain is producing both diversity and affinity. And we can tell the selection people to make the antibodies from this mouse strain only in future. In order to capture the full diversity of the antibody panel, we combined the sequence information with the binning data. And we achieved this by using a custom-built BMS tool that can perform multiple sequence alignment by bin. And the multiple sequence alignment by bin can help us harnessing the full diversity of the antibody panel, and can also help us choosing the sequence with fewer liabilities upfront, and also providing a very good QC for our binning assay. In summary, with the combined kinetics, affinity, and binning data, we were able to understand affinity maturation, we were able to understand performance of different transgenic mouse strains, we could explore epitope-specific binding characteristics, and we could give all the results to the selection technology people. We later expanded our studies by more cell binding assays, some more biophysical and biochemical characterization, and finally picked an optimal panel of diversified leads. So that's all I have for today. I would like to thank a lot of people involved in this project, people from the antibody generation group, people from protein engineering group, protein purification group, and bioinformatics. And special thanks to the Carterra team for their support and always promptly answering our questions and fixing instrumental issues time to time. Thank you so much.
Dr. Matheru, that was a great presentation and a wonderful way to start off our webinar. So thank you for providing us with some really excellent data. Before we move on to Noah's presentation, I want to remind the audience to submit their question for both of our panelists at the end of the webinar. Just simply type your question into the Ask a Question tab on the right-hand side of your screen and hit submit. All right, next presentation is up. Noah, the floor is yours.
Hello everyone, my name is Noah Ditto, and today I'm going to be talking about Carterra's LSA instrument, and in particular how this instrument accelerates therapeutic antibody drug discovery. It's no secret that therapeutic antibodies dominate the commercial drug space. Antibodies bring transformative medicines to patients, even cures in some cases. Additionally, antibodies provide opportunities for biosimilars, companion diagnostics, as well as reagents. Collectively, what this says is that a diverse, innovative, and robust clinical pipeline is really essential to maintain this current trajectory of market growth. While the previous slide highlighted commercial success stories for therapeutic antibody drugs, it can't be understated that drug discovery still remains a costly, tedious process that has high levels of failure. If we look, for example, at phase 1 drugs, only 20% of these molecules will achieve market approval. And on average, it will cost about $1 billion and take approximately 12 years from bench to market. Interestingly, studies in this area looking at root cause of failures have identified understanding mechanism of action as a critical parameter in order to predict clinical success. The discovery and development of therapeutic antibodies remains a highly competitive environment. In these examples for PD-1 and PD-L1, we can see the high numbers of therapies and approaches being used with these two particular targets in mind. Mechanism of action is critical in leveraging the outcomes of these trials to achieve commercial success. This data also highlights the need to maintain clear intellectual property in a very crowded commercial space. Part of the beauty of therapeutic antibodies is the diverse way in which their mechanisms actions can achieve therapeutically relevant outcomes. Blocking, targeting, cytotoxicity, as well as signaling mechanisms can all be significant means in which therapeutic outcomes are achieved. What's really interesting about this is there presents a number of different antibody attributes that must be identified and characterized in all these candidates. The LSA is unique in that it finally brings to fruition the concept of high-throughput SPR. In the early stages of drug discovery, historically, SPR has been relegated to a low-throughput assay format that often restricts the amount of candidates that can be screened for real-time binding parameters. These parameters can include kinetics, affinity, and epitope that are critical for informing mechanism of action. With the advent of Carterra's LSA, SPR now can perform assays orders of magnitude above previous platforms and shift the role of SPR to stages of drug discovery previously not attainable. The LSA's 384 ligand capacity allows epitope binning assays that can number nearly 150,000 interactions in a single run. And this really transforms the binning paradigm, again, to an assay that previously was never approachable in early drug discovery, to one that now allows exquisite epitope resolution, that one leads a selection of really well-informed candidates, and also two, allows companies to secure IP in a very crowded commercial space. The example being the PD-1, PD-L1 commercial space highlighted previously in the slide deck. Other aspects of the instrument that are very relevant for early drug discovery are minimal sample consumption, particularly when resources are very limited in the early stages of candidate development and assessment, and fast and intuitive data analysis packages that enable the user to quickly go through very large data sets and make sense of the information. Applications that lend themselves to the high-throughput real-time binding format the LSA affords include kinetic affinity type assays and epitope characterization assays, including epitope binning and epitope mapping. In kinetic assays, the antibodies are arrayed on the biosensing chip surface, and an antigen titration series is flowed across that surface as an injection. And from this, we develop binding curves where we can assess on rates and off rates and ultimately determine affinities. In epitope characterization studies such as epitope binning, the antibodies are also arrayed on the biosensing chip surface. An antigen is flown across the surface, and then a competing antibody is then passed on top of this complex to assess whether it can form a trimolecular complex or not. These competitive events in this assay determine whether or not the antibodies share or do not share a similar epitope on the antigen. And ultimately, from these, we can build heat maps and network plots reflecting the relatedness of antibodies based on their epitope engagement. Epitope mapping studies typically involve the arraying of an overlapping peptide library derived from the antigen, and then the injections of antibodies across this array surface to identify antibodies that bind peptides. This binding information then dictates down to the residue level the specific sites of binding of the antibodies. What differentiates the LSA from all other SPR-based biosensing instruments is its unique 96-channel printhead device that deposits 96 samples at a time and creates up to 384 ligand arrays using about 200 microliters per printing event. When this array is then printed, the instrument switches over to its single flow cell, which docks on the chip. And this sort of oval-shaped flow cell allows the injections of analytes in total volume of 250 microliters to pass across the array surface. And in a single injection, we measure in real time 384 unique binding events. The combination of these two systems allows for a one-on-many approach that can really leverage screening properties. So if you can imagine an array of antibodies on the biosensing chip surface and the antigen flown across this array of antibodies, there's a tremendous amount of data generated using a very small amount of antigen, and in each injection on a per time scale, we're getting a huge amount of information back. The LSA uses flow-based printing to achieve its array on the biosensing chip surface. In this example, 96 samples are sampled from a well plate and passed bidirectionally across the sensor chip surface, returned back to that well plate, and then the sampler moves to a new location, draws up a fresh batch of 96 samples, and deposits them to a different location on the biosensing chip surface. Ultimately, this system can array 3 or 4 blocks of 96, achieving a 384 ligand array on a biosensing chip surface. Once the printhead has generated the 384 ligand array on the biosensing chip surface, the single flow cell then docks on the chip surface and presents an injection across the array. This injection is passed across in a bidirectional flow manner, allowing a fixed volume of analyte to contact the surface for an extended period of time. The sample deck of the single channel device allows for 2 plate positions, accommodating a 96-384 well plate, as well as a sample block that allows 50, 15, and 1.5 mil tubes. In the upper graphic shown on this slide is a representation of the array that the printhead generates on the biosensing chip surface. The vertical pink rectangles represent actively printed locations, and the horizontal blue boxes are interspot references that the software assigns to the array. When 1 block of 96 is printed in the upper panel, 3 additional blocks of 96 are then printed interstitially in order to create the 384 ligand array. Once the array has been created, the single flow cell docks onto this array and injects the analyte across it. The benefit of having a single analyte presented across the 384 unique binding locations is that a very small amount of antigen, for example, enables the measurement up to 384 binding events plus 4 additional 48 interspot referencing events. An antibody's epitope is a fundamental property that is critical in influencing its mechanism of action. Epitopes are innate. They cannot be predicted by in silico techniques, nor can they be engineered. Ultimately, they must be selected for from large panels of antibodies. Additionally, epitope is critical in securing IP, particularly in crowded commercial spaces. By understanding mechanism of actions of antibodies, they can be combined to produce superior therapeutic products. High-throughput epitope binning is one method of divining the epitope landscape of a diverse antibody library, and combined with functional data can identify very much biologically relevant epitope clusters. Ultimately, this uses epitope diversity as a surrogate for functional diversity. On the LSA, epitope binning is first performed by arraying a panel of up to 384 unique antibodies onto the biosensing chip surface, and then, for example, in a classical binning assay format, injecting the antigen across this array, binding to each of the antibodies on the surface, followed by injection of a single antibody to assess whether the antibody can bind the antigen in the presence of each surface-bound antibody. On the left-hand panel, we see sensorgram views of data generated on the LSA showing first the capture of antigen, followed by injection of an antibody across this captured antigen to determine whether or not it can form a trimolecular complex. In the software, control injections of antigen followed by buffer are highlighted as blue, and antibodies that sandwich in the assay are indicated in the upper half of the left-hand panel, while antibodies that fail to form sandwich are indicated in the lower half of the panel. In this particular case, we have an instance of an antibody that is being displaced, whereas the antibody in solution disrupts the surface-bound antibodies binding of antigen and leads to a faster decay rate than would be observed with the antigen bound to the antibody on the surface alone. Comprehensive epitope binning experiments can be quite large in size, but the LSA provides a very simple and straightforward way to run a 3D4 by 3D4 competition event on a single chip using very small amounts of antigen and very small amounts of antibody. Ultimately, there's powerful software tools at the user's disposal in order to make sense of the large amounts of data. With its real-time binding data, the LSA allows for high-throughput binning assays with exquisite epitope resolution. In the right-hand sensorgram panel, we see different instances of antibodies performing displacement-type behaviors against an antigen bound to an antibody located on the biosensing chip surface. This displacement data is organized into a network plot which shows competitive relationships among antibodies and also shows that the displacing antibodies co-localize within a certain grouping, very similar in their spatial relationships, suggesting that they localize on a similar epitope of the antigen. Using the same epitope network plot identified in the previous slide, we can take what on the left-hand side is the default coloration of the software indicating discrete bins of antibodies and add in, for example, ligand blockade characteristics for all of the antibodies. In doing this, the user simply has to paste in a list of values from a spreadsheet source and call them up in the software to be represented. So not only can attributes such as ligand blockade be represented, but also other features such as affinity, developability characteristics, or even expression levels can all be represented in the context of epitope, making candidate selection and stratification much simpler. In another example of using orthogonal data to further inform the epitope clusters, here we have an example of library sources of antibodies being represented on the network plots. This data demonstrates the versatility of epitope binning experiments in order to not only identify and cluster antibodies based on similar engagement of epitopes, but also to understand the diversity present in different library sources as it relates to antibody outputs. In many instances of infectious disease, the human immune response addresses pathogens by targeting multiple antibodies against them. This mechanism can be harnessed to develop therapeutic cocktails using the same process. When these B cells are isolated from humans, they can then be competitively epitope binned by developing a diverse panel of antibodies that target different sites on the antigen. And this is a key enabler because sequence alone is not sufficient to predict epitope diversity. In the development of therapeutic antibody cocktails, high-throughput binning can reveal the epitope landscape of a human's immune response. Using this profile shown in this broad heat map here with neutralization information layered on top of the data, a few key clones can be used as benchmarks to probe a larger library. Ultimately, this neutralization data can be used to understand the functional space relative to each epitope. From this large-scale binning study, 19 bins were identified. Of these bins, multiple were found to be functional. Interestingly, some of these rarer bins showed exceptionally potent neutralization. Ultimately, the design of this broadly neutralizing cocktail is well informed by the data described in this epitope binning exercise. While the LSA is not the only commercially available biosensing instrument that can perform epitope binning assays, it's far superior in terms of sample consumption as well as ease of use and runtime. In this example shown here, using similar instruments, running a large-scale binning assay, assays runtimes would approach at least a month and use milligram requirements of antibody, while the LSA would require a mere 5 micrograms of antibody and about 2 plates and an overall runtime of 6 days. Despite the high throughput capabilities of the LSA, kinetically speaking, the instrument benchmarks very well against the Biacore AK, for example. In this panel shown below, we can see nearly identical kinetic behaviors for a panel of 6 different antibodies run on both systems. Looking in more detail at this benchmarking exercise, we can see that the LSA's kinetics in terms of K on, K off, as well as calculated affinity are in excellent agreement with the Biacore AK. This kinetic rate and affinity constants remain within twofold across both platforms, testing a group of 32 kinetically diverse antibodies. In other words, antibodies with very slow on and off rates, as well as antibodies with very rapid on and off rates. These LSA measurements additionally include mean plus standard deviation for the 8 to 12 replicates included per mAb, and this is owing to the capacity of the array that the LSA affords, which is much different than most other biosensors that have very limited ligand capacity and ultimately typically preclude the inclusion of replicates in the assay. The 384 ligand capacity of the LSA array allows the user to design experiments with specific objectives in mind, including scouting for surface density conditions, as well as the inclusion of the previously mentioned replicates. In this particular data set, we see that for a given antibody, we have very excellent agreement for affinity measured across multiple replicates, a total of 12 in this example, providing high levels of statistical confidence. In terms of inter-assay reproducibility, KD values are shown here for the LSA and the Biacore AK. On the left-hand panel, the 30 antibodies screened by the LSA show excellent agreement amongst themselves with the 8 to 12 reps per interaction, showing very tight error bars associated with each of those measures, and overall agreement. On the right-hand side, the Biacore AK data has a very limited set of antibodies owing to its limited throughput and the absence of any replicates and measures of statistical significance due to this limited throughput. By virtue of its sheer levels of throughput, the LSA is disrupting antibody analytics. Throughput levels on the LSA are up to 50-fold higher than the Biacore AK. Minimal sample consumption is achieved via novel microfluidics that allow the one-on-many injection routine. Additionally, assay setup is straightforward and simple. As previously discussed in this presentation, kinetics data are high quality and reliable and are within twofold of the Biacore AK. The analysis software on the LSA is purpose-built to allow detailed kinetic and epitope characterization. All of these strengths allow the LSA to shift the role of SPR upstream in drug discovery to a point where it can actually impact library to leads triage. The LSA enables high-throughput epitope binning studies that can clearly elucidate the epitope landscape quickly and with a resolution that allows for differentiation of antibodies via their mechanism of actions.
Thanks, Noah. That was some really great information and a wonderful way to highlight the power of the LSA platform. So before we move on to the Q&A session, which is coming up in just a moment, I want to remind the audience one more time to submit your questions for our panelists so we can have a really great and lively discussion. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. We'll try to get to as many of your questions as we can. All right, I'd like to thank those audience members that have sent in questions already, but for those of you who are just thinking about it, don't hesitate. Type in your questions, send those in, and we're going to begin the Q&A in just a moment. Bear with us for just a minute while we switch over to the Q&A session.
All right, everyone, let's get to the questions that we have from our audience. Our first question is going to be for Zimple. Zimple, one of our audience members would like to know, what factors do you consider when planning a capture kinetics experiment?
Hello to everyone who's listening. Thank you for joining. The capture kinetics, like it depends on what your samples are. You are doing the capture kinetics for the purified or unpurified samples, and also like the concentration of the sample inside the crude sample, like supernatant, what the titer is. So depending on that, you choose your sensor surface. In my case, I use the HT-30M chip. It's a low or medium density chip provided by Carterra. So it depends on how much ligand you want to capture. So that is a very important factor. And another, the good thing about LSA that you can choose a wide range of concentrations, and because there are so many spots, you can play with it. So the concentration of antigen you want to study is also important for planning the capture kinetics experiment.
All right, great. Thanks, Dimple. Noah, we have a question for you. One of our audience members would like to know if you can run crude samples.
Yes, hi everybody. That's a great question. So short answer is certainly you can run crude samples. The LSA actually lends itself to crude samples very well. So on the printhead side, where we typically array whatever our therapeutic of interest is on the surface of the sensing chip, there's no valves in line, making any chance of clogging or other issues very minimal. So the system is highly flexible in terms of robustly introducing a complex sample, like a crude hybridoma supernatant, onto the surface of the chip without any ongoing issues and staying up and running. Additionally, the system, the printing kind of profile that I'd shown in my slides, where we do a bidirectional or multipass flow back and forth across the chip surface, is really good for enriching really low expressing soups. So you can get really, really good assay sensitivity, some of the best you can get out there on any biosensor with our system. So in terms of screening crude, the system is robust in handling the sample, as well as provides an excellent opportunity to really drill down on those low expressors and not miss them during the screening process.
Great, thank you, Noah. Simple question for you. What are the antibody coupling conditions onto the HT-30M sensor chip? What charges would you make in order to couple antibodies to a different, what changes would you make in order to couple antibodies to a different density chip?
So this chip is a polycarboxylate hydrogel matrix, a linear matrix. This chip is provided by Carterra. And so the running buffer we used was a low pH, like the MES buffer of pH 5.5. And the antibodies, for antibody pre-concentration, we used the acetate buffer of low pH 4.5, and then used the cross-linking chemistry that we use for Biacore experiments as well, the EDC-NHS, where EDC is a zero-length cross-linker that could cross-link the antibody samples onto the surface and making an amide bond. So it's like a covalently coupled antibody samples onto the chip. And then you activate with the EDC-NHS, and then you put the antibody, and then you block the backfill the non-specific sites as well with ethanol amine. So these were the standard chip coupling conditions we used for the HT-30M chip, which is like low density or medium charge density. But if I have a different chip, like with a dextron matrix, or and I want to couple more antibody samples on it, I would vary the concentrations of the antibody depending on the chip capacity.
Great, thank you very much. Noah, back to you. How long does it take to process and analyze the data?
Yeah, so it kind of depends on the type of study, how many samples are in the study. But if we take, for example, a kinetics assay, I would say for a single antigen screened against a panel of 384, say, antibodies, so this is a quick run, relatively speaking, for us. And this would probably take an analyst who's familiar with the software probably about an hour or so to achieve a fully processed data set. So that's 384 affinities in about an hour, analyzed and ready to sort of log into a notebook, if you will. In terms of binning, it's probably a little bit more. So if we did a really robust, 384 by 384 binning experiment, it's quite a bit of data, 150,000 plus interactions that are measured. I would say a solid probably 4 hours of time would be necessary at a minimum to go through that data and really understand it. There's just a wealth of information in there that does take the analyst some time to go through and kind of understand in detail.
All right, and stick with me, Noah. I have a couple more questions for you.
Sure.
An audience member would like to know, for the epitope binning, can you explain what you mean by 384 by 384? How is it not 384 by 1?
Yes, yeah, so that's a very astute observation. So when I demonstrated kind of how the fluidics work in the LSA, we do build an array of 384 on the surface, and then we dock our single flow cell on top of that array and run one injection at a time. So it technically, within a cycle of an experiment, it is correct to say that we are running a 384 by 1 sort of profile. But the idea with those experiments is we repeatedly compete all the antibodies that are represented on the surface as solution phase analytes across that array. So we would do up to 384 analytes passed over the same 384 ligands on the surface. So that's where we use the nomenclature 384 by 384.
Alrighty. And our last question, how do you choose an appropriate assay format for epitope binning?
Yes, really good question. So really the valency of the antigen drives the choice of assay for epitope binning. And in particular, if we have a multivalent antigen that's, say, a dimer, it's complex to run a classical sandwiching assay where we would have an antibody on the surface followed by an injection of the antigen, and then coming in with an antibody on top of that to see whether or not they can form a trimolecular complex. The issue is we would have two identical epitopes represented in that dimer, and effectively we could see sandwiching, which would be false, just owing to the nature of the valency. So if you do have a multivalent antigen species, the assay format of preferred choice would be to do premix style binning, where we mix the antigen in the presence of a molar excess of antibody. So we saturate all the binding sites on the antigen. So whether it's a dimer, trimer, or some other higher order species, we bind all those sites up, and then we flow that entire mixture across the array of antibodies to assess whether or not they can interact with a surface-bound antibody, i.e. whether or not they have free and uncovered epitopes.
All right, great. And we have another question for Zimple. Zimple, what is the purpose of putting related samples or replicates in the binning set?
Yes, so we had a lot of replicates and related samples in our binning set because first we wanted to validate the LSA for binning, and also we wanted to QC our binning assay as well. So if your replicates or related samples are showing similar blocking, non-blocking profile as which is expected, then your the binning assay went well, and you can QC your binning assay based on that. But if the replicates are showing different behaviors, if they are put at the beginning of the assay and the end of the assay, and if they're showing a different behavior, then there is a red flag. So there's something went wrong with your experiment. So we wanted to QC our experiment, QC our binning, and that was the purpose of putting the replicates and related samples. So it went very well.
All right, great, thank you. And it looks like we have one more question for Noah. Noah, one of our audience members would like to know, are there limits on buffers that can be analyzed, or cell culture media, biological fluids, etc.?
Yeah, so kind of like what I was mentioning before, the LSA does really lend itself to analyzing crude samples. So there is typically no limitation. The only basic requirements we have for sample prep prior to introducing into the LSA would be to do a really hard spin to pellet any cell or debris material, as well as, or alternatively, you could do a filter type approach, passing everything through a filtration membrane to remove that same type of material. But otherwise, that's the only limitation. Really, any fluid that doesn't have bulk debris in it is suitable for the system.
All right, great. Thank you very much, panel. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be available on our site for up to a year at www.genengnews.com. So if you missed any parts of it, you can watch it again, or feel free to forward the link to any of your friends or colleagues, which we always recommend. I'd like to thank Zimple and Noah again for their very informative presentations, and I'd like to thank you, the audience, for your attention and very thoughtful questions. And a very special thanks to Carterra for sponsoring this webinar. Hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now.